Okay, it's on. So, thanks Marcel, and thank you for the committee for the invitation. It's a great honor to be here today to speak to you. So, from the content of the meeting, of the speeches, I noticed that this is very much about indoor air, and I hope I will talk more about the buildings, the structures, and their significance. So I hope that will be of interest to you as well. So, well, my background is in the academia. I used to work for 10 years with microbes and DNA and molecular techniques, and then I moved to so-called more practical work. So I do condition investigations and project managing and consulting and teaching and etc. So, topic is molecular tools and microbial ecology of buildings, practitioner's view. So. In general, when we talk about the microbiology of the buildings, our goal in the academia as well as in the practical side is to understand the microbiological processes in building structures and into the indoor air, to indoor environment, to understand their potential health effects, among other things. Of course, we're also interested in their effect on the building structure, in other ways as well, but health is very, very important. So in practical work, this means more or less condition investigations and also indoor measurements, indoor investigations, but mostly this so-called condition investigations where we define the condition of the building and then do risk assessment of the potential health risks associated with the condition. And after that, of course, our goal is to choose good remedial actions to improve the situation if there is a considerable problem. But of course, in renovating, the principle is always remove everything that you can, but it's not always possible. So we also need information to support actions on just controlling the emissions, and after also the renovation has been done, controlling the success of the remediation. And in addition to using the information of the microbiology in buildings in the renovation and investigations, we also need the information in new building design, in engineering design and architectural design. Building physics is one field that I'm very much involved nowadays, and it's highly important. So, molecular tools, what's that about? I will tell a bit about the toolbox that is available in the academic side and on the field side today. So, we are in molecular biology, we are targeting macromolecules in cells. That's DNA, proteins, and RNA, and so on. But mostly we're talking about DNA in the methods we are speaking with this field. So we target the DNA, the hereditary material in all organisms' cells, which contains information about the life form in question. So we target so-called barcode genes, which can be used for either detecting a specific organism or identifying various kinds of organisms, because those gene regions contain phylogenetic information of the organisms present there. So these methods, the key in this method is that they are independent of the cell viability. Traditionally, we've been cultivating the microbes from the environment, and that always selects for the viable and the fresh ones in the sample. But when we use DNA, we can see everything, so to say. Dead ones, like half-degraded ones even, but not just the viable ones. A lot in the academia concentrates nowadays on so-called microbial community sequencing. That means that we extract the whole DNA of, for example, dust sample or air filtrate sample, and then we screen all microbes in the sample by reading their DNA, this barcode. But there are also other methods that can be used, but sequencing, this whole community sequencing is very, very big thing today. So these methods have been they have been developed like 30 years ago, so it's nothing really new. The first inventions were the DNA extraction and PCR, which is polymerase chain reaction, which is the very key method for amplifying DNA. These interesting genes from the material, they were invented in 80s and 90s. And in building studies, these methods have been used 20 years or so, from the 1995 onwards, and increasingly popular and widely used. One really significant event was the invention of this so-called next-generation sequencing methods, which are very, very, you call them also highly parallel methods, because you can produce lots of information, lots of data. You can deep mine the samples, get DNA sequences, these barcode sequences of hundreds or thousands of organisms, while previously with the more laborious techniques we could get, for example, tens or hundreds of sequences from a sample. So these so-called next-generation sequencing methods have made it much more efficient than it was previously. And Jordan Pecha will be talking about these methods more today in the Sloan session, so if you're interested, come to listen to him. And that new method has induced some sort of explosion in the field of molecular microbial ecology and also in building sciences. So it seems that now we are trying to sequence everything, all environments, including human beings inside and outside and in the built environment and atmosphere, oceans, all kinds of soil types. So we extract taxonomic phylogenetic information of the bacteria, fungi, viruses, archaea in the samples, in the environments. And after going deep into the DNA, which tells about who is present, the next methods will deal with the functionality of those organisms in the samples. So we will dig into the RNA and the metabolome, the metabolic product, or all kinds of emissions and metabolic products that are produced by these microbial communities. So I will go through quickly about what has been found thus far using these new fancy methods. So in a couple of words, if we're talking about undamaged buildings with no moisture damages, we find so-called core species, a couple of tens of species, which are more or less present always, or not always, but in most samples that we take. But in addition to that, we see a huge diversity of rare species. For example, here we can see the general diversity. For bacteria, we take one sample, we see on average 50 species. Previously, when we did cultivation, it was a couple of species, and now we see like 10 times more diversity. And with fungi, it's a little bit less, but anyway, considerable diversity. And anyway, it goes so that the outdoor air is always more diverse when we compare with indoor air. And then we have these rare unknowns. If we look at the how it looks when you plot or put them in a chart, we see that we have had 55 samples here, and we have only a couple of so-called OTUs, which roughly correspond to a species. Only a couple of species occur in high number of samples, but then there are plenty of species that occur only in a couple of samples. So there is this vast diversity of rare species which always keep on coming. And then for fungi, we can say that the fungi in indoor air, the mold, the yeast, they come from outdoors in a normal situation. Adams and colleagues put it nice that sinks are sources, other surfaces are sinks. Sinks are wet environments, and these make a difference, but all dry surfaces are like sinks. These microorganisms deposit on the surfaces, and they don't do much like active stuff there. They just sit there and die away if it's dry. And so this is how it looks when you look at the diversity of fungi globally. You see that these samples have been arranged according to the latitude. Here is the northern parts and the southern hemisphere and the tropics. We see here are, if you can see the names, these are the most common species present in the samples. We see that they are present like globally, more or less globally, these certain types, but we have a difference in different geographic regions. But for bacteria, they come from us and the pets. Living beings, we shed millions of bacterial cells every hour into our environment, and we are able to, if we go to a sterile new building, it's a couple of hours and days, and then we have created our own like fingerprint or imprint of our personal microbiome into the space. It happens really quickly, and it depends on your personal gut microbiota and skin microbiota, and also your habits and tendencies, how you run in and out and have pets or cook food and so on. But bacteria come from people. And here we can see one example of that, how this is mattress dust samples and flora samples. And here we see the species that were human-derived, and this is the others. And this was in mattress dust. It was even more significant, the amount of human-derived bacterial species. And in undamaged buildings, wet surfaces, as I said, sinks are sources. We can see an enrichment of osmophilic and biofilm-forming bacteria and fungi, for example, in drains and sinks and shower curtains and like surfaces that are wet often. So these are the sites where microbes proliferate and really can produce something in normal building. Some other findings. These are not very surprising, since most of these findings have been made in 1880 something, like more than 100 years ago. But anyway, these findings have been lightly verified and verified with a little more additional information with the new methods and new studies. So we know that dust resuspension, this significantly contributes to the amount and types of airborne microbes. We also have noticed that when we have densely populated spaces, we see more bacteria that are coming from the people. And if you open the window, you bring outdoor air, which is like physically different from the indoor air. It's cooler usually, and the relative humidity is different, and it also dilutes the indoor communities of fungi and bacteria. And it's also obvious that when you have rooms that are close by, they contain more similar bacterial and fungal assemblages than the distantly located rooms. And what is interesting is that when you have an old house, old home, you've got more diversity, especially on the fungal side. So what is the use for me when I go and investigate a building and try to find what's causing indoor air problems there? So I wish that the scientists would give me tools for direct health-based exposure assessment, but unfortunately there are no such tools. We don't have anything specific that we could measure that would function as a good proof for a health risk at the moment. So we need a holistic approach to investigate or approach the health thing. But building diagnostics, that's what we are doing all the time, and also with molecular methods today. So we use so-called qPCR, quantitative PCR, which is a method different from the sequencing thing. So that's a method for targeting specific fungal or bacterial species or groups. We can nicely quantitate them very reliably. So that is one tool that we are using, and then we get information about unexceptional conditions, unexceptional microbial sources in the building. Essentially, there are mold damages and mold growth in the structures. There is this ERMI tool, and other alternatives need to be a little bit fixed locally if you want to start using them. They can be used for material samples to identify or verify microbial growth or damages there, or to sample air and verify unexceptional situation with the airborne microbes. When you deal with materials, there are some problems with old organic materials because they contain loads of microbial DNA by heart, because of their nature. They are usually organic insulation materials and so. The advantages of the DNA methods are speed. The speed, because if you cultivate microbes, you need 2 weeks of incubation time, always 1 or 2 weeks. But the DNA methods, they take 1 or 2 days. So it's very quick, and it can be much more sensitive, and also like wider. You can target a wider assembly of microbes if you want, using those methods. But because we have to know what we are looking for, and when we are only targeting like maybe 20 species or 30 species, we have no idea what else is there. So this qPCR method is such that you can only find what you want and name what you are looking for. Also, if you want to widen the species coverage, if you want to see like many tens of species, it becomes very expensive and not good for practical situations then. And this other method, I talked about this community barcode sequencing, whole community sequencing. It produces too much useless information from our perspective, because we don't want to know about the human-born bacteria. We don't want to know about the rusts and mushroom spores that come from the outdoor air. We want to specifically detect the signs of abnormal microbial conditions in the buildings. But if you think about the advantage that could be later coming to the practical field from the academic research, we live in the hope that all these molecular investigations, they will just add to the basic knowledge about what's happening in the buildings. And then we will get like a better understanding of the exposures and disease mechanisms, well, from the general understanding. So if there is something that we haven't found yet, new taxa, new interesting taxa that are producing new metabolites, mycotoxins or volatiles or something, and then we can, if we detect something interesting, we can target to the specific metabolic genes or directly to the metabolites. So loads of new information is about to be coming from that side of the research, and then as a result of that, we might develop better diagnostics tools for building investigations using these methods. So in my dream world, we would have this some sort of community sequencing or this metabolomics community DNA combinatory method where you could extract fingerprints of abnormal situations of various types. But I don't know, that's years away from the present situation. If we could extract a health signal from there or like damage signal from the big amount of data that we can get today with the new methods. So today the research is largely it's concentrating on when we talk about the publications that have been put out on the field. They concentrate largely on indoor surfaces. This is a representation of the mechanistics of indoor microbiomes. There are green sources, and then there are sinks and dispersal vectors and environmental factors. And there is soil, vagina, gut, skin, pets, and outside air as sources. But from my perspective, these are not interesting. Well, of course, they contribute to our living environment, but the immediate adverse effects are usually related to what's happening in the constructions and not on the surfaces in the buildings. So when we're looking for something interesting, like from my view in a building, we're not interested in, of course, we look at the surfaces to find signs of something that's happening inside there, but the hotspots, the microbial hotspots, the places in the building where you have conditions that really support proliferation and growth of the microbes on active things, they are mostly inside the structures. We have layered structures in the building envelope mostly today. Of course, we have massive, or in old buildings, we have massive brick constructions and so, but increasingly we are talking about layers, structures where you have drywall, gypsum board, and mineral wool insulation, all kinds of insulation materials, and many types of layers which nicely hide things inside. So, in a typical housing, like a small house, which is built in the Scandinavian way, with insulation in the envelope, we have these hidden places. Of course, these are like they develop with time. So in old buildings we have almost always some microbial growth, even if it's not like a really problematic or really bad condition. But there are sites that tend to get some water in and develop something additional in there. So these sites under the slab, under the ground floor, are always they're like obvious sites. If you have too much moisture in the crawl space, it's pretty obvious. And then like pipe leakages, they are quite obvious, and roof leakages, obvious sites. There is a dam that forms in the springtime when there is heat is leaking out of the roof and creating ice there, and it's then leads into roof leakages more easily. They create something here, and then you have leaking windows and leaking these moisture barriers in the wet rooms, and all kinds of sites. You may have wicking damp from the ground that creates problems on the flooring, and if there is insulation below the slab, then it's an additional very common place. And all ground-facing structures we have there, cellar walls, especially if there are insulations, they are the sites that we are looking at. From my view, I am a microbiologist with the knowledge about building physics and building techniques and the constructions. I find them increasingly interesting. It's really fascinating to find out what kind of niches, microbial niches, the sites of like community development and microbial living environments you can find there. So instead of looking at surfaces, we should be looking at the structures, as we probably mostly, if we go there, we are doing it. And talking about the niches, in buildings are like the conditions are always they make some sort of equilibrium between the indoor conditions and the outdoor conditions, and then there are various types of materials in various parts of the buildings. So taking an example of this cellar wall, there is very, very nice study by Nunes and colleagues about this certain structure as a microbial niche, how the external conditions, relative humidity, salinity, pH, and indoor conditions create this specific environment into the structure. And this group found that these various layers in the structure, they tend, like on average, develop certain kind of microbial communities. And this is what I find interesting. I don't like reading that, okay, when you open the window you get like outdoor mold spores in, but this is something that is very close to like my daily life, still on the academic side, but potentially useful. But thinking about the structures, why should we be interested in the structures? Because they are not in the indoor environment. If there is some hole somewhere which is dirty or creating some growth, why should we care about that? But if we think about the building, the conditions, at least in the cold climate, we have under pressure, more or less. We have to have an under pressure of some sort in order not to push the moist, warm indoor air to the structures where it would condensate and create big problems. So we have constant under pressure. Of course, this whole package goes the other way around in the tropical climate. There you have to have a overpressure. Anyway, we have an underpressure, and if you think of a house, like your house or my house, typically, well, it could be small, could be big, but an example, 100 square meters, and volume of 270 cubic meters. The average leakage rate in an old house could be 4 changes per hour, so 4 volumes per hour is coming through the envelope, not through opened window or through some control thing, but through the envelope. It's infiltrating. These 4 changes, if you compare it to the averages in UK, Canada, Sweden, it's not a good quality, but it's pretty normal. And that kind of leakage rate in heavy under pressure, it gives you 1,000 square meters of air per hour. And in normal under pressure, 5 pascals or so, which is like optimum, you still have 30 to 50 square meters per hour infiltrating through the envelope. So thinking of, usually we have older buildings are more leaky, new ones are less leaky, and we think of the average age of the building stock. I hope some of you can see something of this and you can spot your own country. We have information of various European and US and Canada countries, and for example here we have Finland. This is the decades where the buildings were like built. This is before 1919, and then there is the wartime, and after wartime. So we can see that in almost all countries we have a peak during the wartime. That means that the buildings are over 50 years old, many of them, and anyway over 30 years old, which means that, well, we have microbial niches, some activity in the structures, and we also have the structures tend to be quite leaky. Some examples, I don't know if you recognize, of course the structures vary from country to country, what the building practices have been. These are the Finnish examples, some Finnish examples of structures. I've painted the insulation layers, which usually contain some interesting things microbiologically. I painted them red, and we can see that we have this interesting site. This is quite modern, this concrete sandwich element building's external wall, and then we have these ground-facing old structures and wartime, well, after-war structures where you have various types of organic insulation materials. Nowadays it's mineral wool, more or less, or EPS. Like interesting sites. Well, several interesting sites to look into. Of course, these vary from country to country, but we in Finland, we did not invent these structures. So in addition to the structures, we also can have like obvious sites of or sources of microbial activity, all crawl spaces, underground slab going these channels for hot water pipes and all kinds of nasty sites that can harbor like vast microbial activity. And the pressure, it's not, I don't really remember if I read about the pressure differential in most of the papers that have been published concerning indoor microbiomes. But this is really important. We have buildings in Finland that are in bad condition and people are more or less happy. That might be, or often is due to the rather balanced ventilation. And we have buildings with minor problems where there are or minor structural problems with unhappy people because there is heavy under pressure. And in this figure we have an example measurement from a typical building where the HVAC system is mechanical in an exhaust, so balanced ventilation, which had been adjusted a month or so ago. It was supposed to be completely okay, but we see that it's constantly going below zero line. The green is balance, zero pressure, and it's not easy. In practice, it's not easy to keep the pressure balance in a building, even with the balanced or mechanical in and exhaust ventilation. Yeah, so I'm running out of time already, but anyway, we have nice examples already from the cultivation studies about the microbial diversity, the species that tend to occur in certain materials. For example, these correlations with the fungal mold genera and various types of building materials. So there's plenty of things we know already about these things. But there is still more to do. From my view, what we could do, you could do, is like deep characterization of these interesting microbial niches, these common problem sources. And in addition to the microbes, to the taxa, we should look into of course, the conditions that affect to the development and the activity of the microbes there, but also the outcome of the microbial activity, the emissions, the metabolic products that they are producing. And this is where this chemistry should combine with microbiology, and when choosing the site, what to investigate, the building physicists and building scientists should combine with microbiologists. And also a very interesting thing is aging old materials, which are not really looked into the present studies. So we always, if we go investigating the community structures or the development of microbial communities, we have a model in the lab, and it's usually a couple of months, not much more, that it's followed. And that's nothing compared to the natural environment. We are talking about decades in the real systems. So we should really consider that very, very long time scale and the technical lifespan that we are expecting from the building structures. They should work for 30, 50 years at least. I will skip something. There is a nice example of what has been found using molecular methods in specific environments. This is of very ancient mural paintings, so solid stone-based, brick-based structures, which look like this. So they were looked into by using cultivation. They looked what kind of bacteria you will find there. They found a bunch of specific bacteria that are growing in that kind of materials, but using DNA-based techniques, sequencing, the bacterial diversity looks like this. They're uncultured, unknown, unclassified, things that we have no clue about what they are doing there, what are they. And in addition to those, we have plenty of this diversity on top of that. And in addition to bacteria, which have been investigated and can be investigated using the traditional techniques, they found archaea, which are things that usually we find in extreme environments, but it seems that they are common also in normal environments, and we can find these using the DNA-based methods. Yeah. So one thing to take into account, in addition to looking into the structures instead of the air and the surfaces, is that we should combine with the chemistry, yeah, as I said, chemistry and the building sciences. So in addition to what the microbes are doing there, we can also have like non-biotic action, degradation going on in the materials, and that those should be taken into account when you think of the health effects of what's happening in there. And also we may have fibrous, particulate, other non-organic impurities that are present all the time in these old degrading buildings with the microbial impurities. So to summarize, I put it so that from my view, dirty kids and kids with dogs, we know that they are healthy, but kids in moldy buildings are asthmatic. So this moldy building is one still. This is very like old-fashioned talk, but it's still very valid because it's one of the main factors making people sick in the buildings. So what we have, the molecular tools have revealed an unseen microbiological diversity in the environment, but the focus is on surfaces and not the structures. We have interesting niches in the structures, and they are very important contamination sources. So we don't really understand what's the entity still. And it's not just about the mold. So I want to know how can I avoid it when we design new buildings, and how can we fix the problems. So that's about it. Thank you.